Hello my dear students and welcome back to my channel. In today's video, we will be discussing about the topic mass spectrometry, which is a very important topic from your board exam point of view. So, without wasting any more time, let us move towards the video. So, the topic for today's video is mass spectrometry from chapter number 2 of class 12 biotechnology. It is also one of the ways to characterize the protein, and mass spectrometry is an analytical device which is used to measure the molecular weight of protein or peptide or any other chemical compounds. So, let us quickly see the principle on which this technique is based. So, the principle of mass spectrometry is to determine the molecular weight of proteins. And how it will determine the molecular weight? It will determine the molecular weight by separating the proteins according to their mass by charge ratio, or we can say m by z ratio, where m refers to the mass and z refers to the charge. So, the proteins will be separated according to their m by z ratios. How we will understand later in this video only. So, as it is an analytical device, let us understand the instrumentation or the process of this technique. So, first of all, let us draw the diagram of this mass spectrometry. This is a very simplified line diagram of the process. First of all, we have to draw an arc, double layer it, and in the middle of this arc, we have to draw another angular process. So, the first step of this technique is that the sample is taken in the vaporized form. मीन्स जो सैंपल यहाँ पर यूज़ होता है वो वेपराइज्ड फॉर्म या गैसियस फेज़ में होना चाहिए. अगर वो सैंपल गैसियस फेज़ में नहीं है तो कई सारे टेक्निक्स हैं जो कि उस सैंपल को वेपराइज फॉर्म में या गैसियस फॉर्म में कन्वर्ट कर सकते हैं. वो भी अभी हम आगे देखेंगे इस वीडियो में. सो आफ्टर द सैंपल is inserted into this instrument, the first step is the ionization of the sample. Ionization means to generate ions, because then only we will have the m by z ratio. This ionization process is also discussed later in this video. Now, after the ions are generated, these ions are accelerated, and when these ions are accelerated, then along with the electromagnet present in the instrument as well as the electric field, these ions are deflected. So, the third process is deflection of the ions. And after the deflection, the signals are detected on the detector. So, the fourth step is the detection. And as very low quantity of samples can be used in this instrument, therefore, the signals are also very weak. So, after the detection, these signals are sent to the amplifier where the signals are amplified. After the amplification of signal, the signals are sent to the computer where these signals are processed in the form of chart and they are processed. So now let us see how this ionization step is done. So as we have seen earlier that the first step is the ionization of the sample. Now how this ionization is achieved? So the sample are ionized either by the loss or gain of electron. This is usually done by either electron ejection or protonation or deprotonation. Usually the proteins are protonated, as we all know that the proteins are made up of amino acids, and the amino acids has the amide group which can be easily protonated. Also, this protonation can be done in the proteins where the basic amino acids are more in number. Basic amino acids like lysine, arginine, or histidine, which have an extra amide group. Due to this, the samples may be multiply protonated, means more than one proton can be given in the protein, and therefore after the samples are ionized, the samples are separated according to their mass by charge ratio. Now let us take an example and see how the M by Z ratio is taken out of any protein. So suppose we have a protein which is having the molecular weight of 10,000 Dalton, and after ionization the protein is protonated at multiple sites, means either the protein has been protonated at 5 sites or 4 sites or 3 or 2 or even 1 site. Then how we will calculate the M by Z ratio? Let us see. So this is the formula which will be applied for calculating the M by Z ratio, that is M plus NH bracket close N plus upon N, where M is the molecular weight of the protein, N is the number of hydrogen ions, which means the number of protons which are attached to the protein, and therefore if the protein has the molecular weight of 10,000 Dalton and if it is protonated at 5 sites, then its molecular weight will be equal to the molecular weight of protein plus, as 5 protons are added and each proton has a mass of 1, therefore the total molecular weight of the protein will be 10,000 plus 1, and it, as it is protonated, so it will be having 5 positive charge. Therefore the molecular weight 10,005 will be divided by 5, and therefore we will get the M by Z ratio as 2001. Similarly, if the protein is protonated at 4 places, then its M by Z ratio will be 10,000 plus 4, because this time 4 protons are added. So 4 will be added in the molecular weight of the protein, and the M by Z ratio will be 10,004 divided by 4, which comes to 2501. Also, if the protein is protonated at 3 places, then the value will be 10,000 plus 3 upon 3, means 10,003 divided by 3, which comes to 3,334. Similarly, if the protein is protonated at 2 places, then we will add 2 to the molecular weight and divide the 10,002 by 2, which will come out to be 5,001. And if the protein is protonated at only one place, then the M by Z ratio will be 10,000 plus 1 divided by 1, which comes to 10,001. So, if the protein which we have taken, which had the molecular weight of 10,000 Dalton, then we will get the M by Z ratios of 10,001, 5,001, 3,334, 2,501 and 2,001. So, the relative intensity of these M by Z ratios will be plotted in a graph like shown here. On the x-axis is the M by Z ratio, whereas on the y-axis is the relative intensity. And here we will get the graph like shown here. With the help of this graph, the molecular weight of the protein are determined. Now, students, one more thing. Sometimes in the examination, it is only asked that how many M by Z ratios we will get from a particular protein. They will give you the molecular weight of the protein and the sites at which they are protonated, and you have to find the M by Z ratio. So you have to remember the formula of taking out the M by Z ratio. Now, after the signals have received upon detection, these signals are transferred to a computer which stores and processes this information for finding out the molecular weight of the protein. Now, one more term is given in your book, which is MALDI. Now, the full form of MALDI is matrix assisted laser desorption ionization, and this technique is used in the first step, that is the ionization. Means, as you have seen that the samples which is to be taken in the MALDI should be in the vaporized form. Therefore, there are many methods which are used for the vaporization of the sample, and one of those methods is discussed in your book, which is the MALDI method. And in this MALDI, there is a matrix material in which the sample is mixed, and then, as the name suggests, matrix assisted laser desorption ionization means the sample is mixed with the matrix, and pulsed laser beam is shot on the matrix, and this matrix absorbs the laser light energy and causes the matrix material to vaporize or evaporate, and as the matrix is vaporized, therefore the sample also gets vaporized. This was all about mass spectrometry. Hope you enjoyed the session. If at all you have any comment, please do write in the comment section below.